The success of gene cloning is thanks to the various biological molecules that can be used to manipulate the DNA, especially cutting, joining, and modifying the DNA. So, this session is going to be an overview on the range of DNA manipulative enzymes that is involved in gene cloning. So, let us look at the learning outcomes of this session. So, apart from just cutting and joining DNA, as one observes in the basic steps of gene cloning, where the first step itself is cutting and the second step involves joining, you have, other than these two, other modifications that are also seen in cloning experiments. And this has been possible because of a range of manipulative enzymes. Now, the DNA manipulative enzymes are broadly classified into four groups, and they are nucleases, ligases, polymerases, and modifying enzymes. Other than DNA, there are some enzymes which are also able to help in acting on RNA molecules, and these can also be applicable in gene cloning. There are some enzymes that can be classified into more than one group. That is, some enzymes can act as nucleases and they can also probably act as polymerases. So they have a dual or multiple catalytic activities. Let us first look at nucleases. Now, when one takes or hears of the word nucleases, then it is suggestive of the fact that this enzyme has to do something with the nucleic acids. And what the nucleases do is cut the DNA. So they are responsible for degrading the DNA by cutting the DNA. So effectively, nucleases cut DNA, and the meaning of cutting DNA is basically breaking a phosphodiester bond, because nucleotides are linked to each other through phosphodiester bonds, and that is how you have an entire polymer of DNA being formed. Now, nucleases itself can be again subdivided into two major types. One of them is what is called as exonucleases. As the word suggests, exo means outside. So nucleases, that is enzymes that cut DNA only from the outside. Now, in case of a DNA, what do we mean by outside? It means the ends of the DNA or the terminals of the DNA. So if this is the DNA, then the cutting by the exonucleases are at the ends of the DNA. So, what joins the two strands of the nucleic acids or polynucleotides is the hydrogen bonds, but what holds the entire polynucleotide together is the phosphodiester linkage. So, the cleavage is of the phosphodiester linkages, but only at the ends of the DNA. So if one keeps the DNA exposed to exonuclease for a long period of time, what is eventually going to happen is a long DNA will become shorter over a period of time. Now, there are several different types of exonucleases that are used in recombinant DNA technology or in gene cloning. Just to understand that exonucleases also may have a little different way of functioning from one another. So here is an example of BAL31 that has been isolated from a bacteria. And BAL31 is able to cut the DNA, double-stranded DNA, from both ends such that it cuts both the DNA strands. So you can see that you have a nucleotide that is released from this strand, but you also have a nucleotide that is released from the other strand. And you can observe that happening at both the ends. So therefore, you will have gradually one by one a pair of nucleotides being removed every time there is a cleavage, and it is always from the ends. Now you also have exonuclease 3. This is derived or obtained from E. coli. This has a slightly different way of functioning in the sense that exonuclease 3 removes a nucleotide from the end, but only from one strand of the DNA, not from both the strands of the DNA. So you can very clearly see over here that exonuclease 3 removes the nucleotide from the 3' end of the double-stranded DNA. So it will remove the nucleotide from this 3' end as well as remove the nucleotide from this 3' end. So when it keeps continuing to remove the nucleotides from the ends, what you would observe is that you would have a single strand overhang being formed on the other strand where it is not cutting. So at the 5' end, you will have an overhang being formed. So this is one overhang, this is the second overhang. And from the 3' end, the nucleotides will keep being removed by the exonuclease 3. So very clearly that you can observe over here is this, that for this DNA which is becoming shorter over a period of time because of the activity of BAL31, you will get blunt ends. But when you see over here exonuclease 3 cutting, what you will get is sticky ends, or what you will get is overhangs. So this is one staggered ends, rather. Let us not mention it as sticky ends as of now, but let us mention this as staggered ends, and here you have blunt ends. So therefore it is very clear that within the exonucleases itself you can have exonucleases that are acting differently. Then we go to the next type of nuclease, and that is endonucleases. As the word suggests, endo means within a DNA. So that means these are enzymes that cut the DNA, but not from the ends of the DNA. Anywhere within the DNA the cut can be made. So if this is the polynucleotide, then if this is the double-stranded DNA, then the endonuclease can cut anywhere within. So if it cuts here, you will get two fragments. Then the endonucleases can cut these fragments further, so these can become again smaller. So what you observe over here is that a long DNA can be cut into smaller fragments by the endonucleases. Now, again, within the endonucleases, you will have different ways by which endonucleases can function or act. So, say for example, S1 nuclease. Now, S1 nuclease is specifically a endonuclease that cuts only a single-stranded DNA. So, if you have a single-stranded DNA, then this is more susceptible to cutting or degradation by S1 nuclease, as you can see over here. So it gets cut into smaller, very smaller fragments of DNA as you keep it exposed to S1 nuclease. If there is a double-stranded DNA, the S1 nuclease will not cut unless there is a nick. Now, because of the presence of a nick or a gap in one of the strands, and the other strand where there is a nick, you will find that this is single stranded, and so the S1 nuclease will now cut this strand, and because it cuts the strand you will get two fragments. However, if this nick wasn't there, then the S1 nuclease would not have cut the DNA. Now let us look at another endonuclease, and this is DNase 1. DNase 1 can cut both single-stranded DNA as well as double-stranded DNA, and it can cut anywhere within to give rise to smaller fragments. The third type of endonuclease, which is very, very important and highly used in gene cloning, is restriction endonucleases. Not that these are not being used, these are also being used, but for the cutting purpose in terms of the gene cloning experiment, restriction endonucleases have found themselves or have created for themselves a very special place. Restriction endonucleases, as the word suggests, it is an endonuclease, so it can cut anywhere within the DNA, but its cutting is restricted only to a specific sequence in the DNA. So if the sequence is present over here, then it will cut only at this region. It will not cut here or here. It will only and only cut at this particular site. That is why it is called as restriction endonuclease. Now we go to the next set of enzymes that is also used. So you can always relate ligases to ligation, that is basically joining. And we know that joining is very much a part and parcel of the gene cloning workflow. So ligases join the DNA, which means that they seal phosphodiester bonds. So if you have a 3' OH and a 5' phosphate, then immediately there is a sealing of the 3' OH and the 5' phosphate to form a phosphodiester bond. So therefore, effectively one understands that if you have discontinuities in the DNA, then those discontinuities can be filled up. So if you have a DNA where you do not have one single bond over here, immediately you will have ligases coming and binding and sealing the gap. So this is definitely something that DNA ligase does, and hence you will find that it has a very important role to play during replication itself, where the lagging strand is synthesized discontinuously and all the Okazaki fragments are finally joined to each other by DNA ligase. Similarly, DNA ligase has an important role to play in DNA repair, because once you have the repair done, the final sealing of the repair work is carried out by DNA ligases. But it has obviously another role. So, for example, if you have two different DNA molecules, then the two different DNA molecules can also be ligated to form one DNA molecule. So, this is essentially what is making it very important for the gene cloning method. Exactly the point. That is, if you have the vector and you want to insert the fragment of DNA within the vector to form a recombinant molecule, you can see that this end, suppose this is the DNA fragment and this is the vector end, then these two have to be joined over here and these two also have to be joined over here to form a complete recombinant DNA molecule. So you can see very well that right in the second step of the gene cloning, joining or ligases play a very important role. Then we come to polymerases. So we all know what polymerases do. Polymerases are of various types, DNA pol 1, 2, 3 in bacterial systems, and of course you have myriad number of polymerases in the eukaryotic system. And these polymerases carry out one function, and the function is of adding nucleotides and synthesizing a new polynucleotide by using a template. So the basic reaction of the polymerases is that if it has a template, it will read the template 3' to 5' and using a primer, so it cannot ever initiate synthesis on its own. It needs an existing platform to which it can add nucleotides. So it should always have a 3' OH end, and so that is provided by the primer. So once you have the primer, the polymerase just simply adds the nucleotides, but every nucleotide that is added is complementary to the nucleotide present on the template strand. And that is exactly how a new strand is being formed. So suppose one wants the double-stranded DNA to be labeled. Then what can be done is that a template be used, a primer added, and then the nucleotides that are added are all labeled nucleotides. So what will happen is that the final double-stranded DNA that you will get will have labeled nucleotides. And so this DNA molecule can be identified by that label. One of the best examples of how polymerases are used in cloning or in recombinant DNA technology is the use of Taq polymerase. So the word Taq comes from Thermus aquaticus. So it is a bacteria that is obtained from hot springs, and therefore the Taq polymerase is an enzyme that can tolerate temperature as high as 94 degrees, and Taq polymerase is therefore used in PCR, that is polymerase chain reaction. And in polymerase chain reaction there is a there is a stepwise process of increasing the temperature and then decreasing the temperature and then carrying out the synthesis. So Tag polymerases help in synthesizing the new DNA in the PCR mechanism, and so through PCR also you will get amplification of DNA. But this is just an example of how a polymerase is needed to synthesize a new DNA. So DNA polymerase 1 is used for many applications in gene cloning. So as you can see, if you have a DNA molecule with a nick, then the DNA polymerase 1 will use the 3' OH of the nick and start adding nucleotides. But because the DNA polymerase 1 also has 5' 3' exonuclease activity, it can replace the existing or the old polynucleotide strand and replace it with a new polynucleotide strand, completely new polynucleotide strand from this end. So that is something that DNA polymerase 1 does. So DNA polymerase 1 acts both as a polymerase as well as a nuclease. So you can put this under the class of polymerase, also under the class of nucleases. So how does DNA polymerase that 1 therefore help? It helps in nick translation. So as you can see that it is not just filling up the gap, but it is also removing the old strand, old polynucleotide and putting in a new strand. Again, you can very visibly see that nick translation or gap filling can be used to label a double-stranded DNA. So it synthesizes and degrades, therefore it acts as a polymerase as well as a nuclease, and this can enable helping the DNA to be labeled. Now let us look at another commonly used polymerase, and that is the Lino fragment. Now the Lino fragment basically is a DNA polymerase 1, but this DNA polymerase 1 has its domain of exonuclease cleaved off. So the first 320 odd amino acids contribute to forming a domain that is responsible for exonuclease activity, and when that 323 amino acids are removed from the DNA polymerase 1, the DNA polymerase 1 will lose its ability to degrade the DNA, but it will only have the ability to synthesize the DNA. So the DNA polymerase 1 that is devoid of the nuclease activity is what is the Leno fragment. And so the Leno fragment can only fill the gap, but it cannot displace the old polynucleotide strand that is present to form a new polynucleotide. So existing nucleotides are not replaced, but yes, the nick can definitely be filled in. So it is used for gap filling, and also it can be used using a primer. It can using a template and a primer. It can just synthesize a new strand. That is also possible by Lino fragment, and so the Lino fragment has been used in many sequencing experiments. Then of course one has reverse transcriptase. As the word suggests, reverse transcriptase is a specialized DNA polymerase that would use RNA as a template and synthesize DNA. So the DNA that is obtained over here is what is called as complementary DNA. And so what is achieved or what is obtained over here is a complementary DNA strand that can then be removed from this RNA, and in place of that a new DNA can be synthesized to form a complete complementary DNA. So what you see over here is very clearly that a new strand of DNA is formed by reverse transcriptase. So reverse transcriptase is definitely acting on an RNA molecule. And so reverse transcriptase enables cDNA formed, and these cDNA forms can be cloned as well. Now we go to a few DNA modifying enzymes that are also used in gene cloning. Alkaline phosphatases, for example, are used to remove the phosphate group from the 5' ends of a double-stranded DNA. So you will find that in a double-stranded DNA, if this is the 5' end, for the other strand, the 5' end is at this. So both this phosphate and this phosphate can be removed to get a structure where you have the ends of the double-stranded DNA devoid of phosphates. You will only have OH. So if one wants to add a phosphate bond over here, this being 5 prime OH, you cannot have a phosphate bond being formed at this point. So one can actually prevent formation of phosphates at phosphodiester bonds due to the 5 prime OH, 5 prime phosphate becoming a 5 prime OH. Then one has another enzyme which is polynucleotide kinase. So polynucleotide kinase can be used to add a phosphorus group to the 5 prime OH. So therefore what was removed by the alkaline phosphatase can be added by the polynucleotide kinase. So you get back the DNA with the phosphates at the 5 prime end. One also has another enzyme that is commonly used, and that is terminal deoxynucleotide transferase. So as you can see, the terminal deoxynucleotide transferase adds nucleotides to the 3 prime end of single stranded DNA or to the double stranded DNA. So when this is the case, as you can observe, as the enzyme keeps adding nucleotides at the 3 prime ends, so this end also is getting longer and this end is also getting longer, what you definitely are getting is a staggered DNA. So these are staggered ends. So this is something that is also very important. So you get what is called as 3 prime overhangs. Now, one can also use topoisomerases as a DNA modifying enzyme simply because topoisomerases can cut and remain bound to the cut ends and then join the cut ends again. So you basically have cutting possible by the DNA topoisomerases and you also have joining possible, and therefore for specific activities the topoisomerases can be used. They may create a 3 prime phosphate and 5 prime OH, and so this typical observation or this typical resultant of its activity can be used for specific manipulations. So let us make the conclusions thereby. Gene cloning involves manipulation of DNA that uses a range of manipulative enzymes. There are several nucleases that are used to cut the DNA either at the ends or anywhere within or at very specific sequences. So this is pertaining to restriction endonucleases. And these cut ends can be again ligated by ligases. Polymerases are used to fill up gaps, carry out nick translation, synthesize complementary DNA from RNA, and this element of synthesis is used in different applications of gene cloning. Modifying enzymes like alkaline phosphatase, polynucleotide kinase, terminal deoxynucleotide transferase are all tools for specific requirements in gene cloning. Thus, the range of manipulative enzymes are an umbrella under which more and more enzymes are being included. This has led to understanding functions of genes, editing genomes, enabling epigenetic changes with the aim of developing a product or an application. Thank you.